So this is the DNA sequence analysis part of the bacterial ID lab. And we're going to do it for sample A from the fluid from the lymph node. So we're going to follow the steps here to open the data output from the sequencer in another browser. I don't think it's showing, but basically what I'm going to do is copy the sequence data and then onto the NCBI site. I'm going to copy and paste it into the enter query sequence. And then I'm gonna make sure everything's okay, and I'm gonna click blast, and this should give me the type of bacteria that it's supposed to be. I'm just gonna wait on the blast search. Okay, so after inserting the sequence, this is what I got. Apparently it's the Bartonella Hensley. So I'm going to share the other screen. So I think I'm ready now, and it's this one. Yeah. I've currently identified the bacteria Bartonella Hensley. So that one's correct. So I'm going to do the same thing for sample B. So this is stool sample. I'm going to do the same thing, opening the data output from the sequencer, copy the sequence data, and then copy and paste it and blast it. Okay, so after waiting for the NCBI website to give us an answer, it went to show that it was E. coli apparently. And due to time's sake, I will be pausing in between because it takes a bit of time for the NCBI website to search for the bacteria. So I'm going to be pausing between the samples. So this one was correct. It was E. coli. Now I'm gonna go for the urine sample. I'm gonna go to the NCBI website and do the same thing. Okay, after waiting for the NCBI to give us results, just like the sample A and for the sample B, according to it, it's supposed to be the Pseudomonas aeruginosa. So this one was correct. That's what the NCBI website showed. So I'm gonna do the same for the other samples. For the sample D, I'm gonna open it and then pause. Okay, so according to the NCBI website, after waiting, the bacteria determined is the Salmonella typhimurium, which is correct because the NCBI website showed that to be. So I'm going to find the sample E. Due to time sake, I'm going to pause because it takes some time for it to search. Okay, so after waiting, the NCBI website went to show that the bacteria identified was the Yersinia pestis, and this seems to be correct for the sample E. And so far, the NCBI website has given us everything correct based on the sequence. So the last sample will be sample F, and I'm going to do the same because it takes a bit to search. Okay, so according to the NCBI website, when I copy and pasted the data sequence. The bacteria that it showed me was the Yersinia enterocolitica, which is correct based on what the NCBI website showed me. So far, all of the samples I identified were correct. From sample A, which was the Bartonella henselae, sample B, which was the Escherichia coli, sample C, which is the Pseudomonas aeruginosa, sample D is the Salmonella typhimurium, sample E is the Yersinia pestos, and sample F is the Yersinia enterocolitica. So that's it for this lab, for the sequencing analysis lab, because I already did all these steps previously. So as you can see, all of the samples were determined.